Welcome back to the deep dive. Today, we're going to be all about proteomics.
Oh, cool.
Yeah, that intricate world of proteins tirelessly working away within our cells.
Right, right.
And you know what? You've sent us some really fascinating research on this.
Yeah.
So we're going to unlock those proteomics secrets today, I think.
I'm eager to dive in. It seems like you're particularly interested in how scientists are analyzing proteins on a large scale, right?
You got it.
And specifically how they're tackling the challenge of protein quantification.
You read our listeners' mind. We've got
Yeah.
this really cool paper. It's all about a new tool called quantms from Nature Methods.
Okay.
And we've got some excerpts here focusing on, well, protein quantification.
Right, right.
So what's so special about this quantums thing? The paper makes it sound like a real game changer for handling these like massive proteomics data sets.
Yeah, it is generating a lot of buzz.
Yeah.
Imagine if you had to analyze a mountain of data.
Oh, gosh.
Manually, that's the reality for proteomics researchers. Until now, that is,
Right.
this paper demonstrates how quantums, which is a cloud-based pipeline,
Okay.
can analyze these massive data sets significantly faster.
Wow.
A game changer indeed.
Okay, so instead of like scientists huddle over their computers, it's like Quantas is doing the heavy lifting like behind the scenes.
That's a good way to put it.
But paint a picture for us.
Sure.
Why should we care about faster protein analysis?
Yeah.
How does this impact us beyond the lab?
That's where things get truly fascinating. Faster, more accurate analysis allows us to tackle bigger questions.
Like what?
Like, how do protein levels differ between healthy and diseased tissue?
Oh, interesting.
Can we pinpoint potential drug targets more efficiently?
Right.
The researchers actually used quantums to reanalyze 83 existing data sets,
Wow, that's a lot.
acting like digital archaeologists.
So they're like digging through old data with these new tools.
Yeah, and they're unearthing hidden gems.
What kind of treasures do they unearth?
Well, they identified 479 proteins that had never been quantified before.
What?
Yeah, one example they highlight is protein Q86SP6.
Okay.
Now, we knew this protein's blueprint existed,
Right.
but we'd never actually observed it being synthesized within the body until now.
Oh, wow.
And with quantums, they found it in 16 different tissues.
Wow. That's incredible. Talk about a breakthrough.
It's huge.
It really highlights the power of this kind of research.
Absolutely.
Speaking of protein quantification, your other excerpts mention something called IBAQ,
Right.
and comparing different methods. Do you care to break that down for us?
Of course, IBAQ stands for intensity-based absolute quantification.
Okay.
Essentially, it's a way to estimate the amount of each protein in a sample, not just its presence.
Okay.
So think of it like taking inventory. You need to know how much of each ingredient you have.
Otherwise, your recipe is going to be way off.
Exactly.
And how does quantums measure up against other methods in this way?
Well, the researchers found that quantums performs incredibly well,
Really?
even identifying proteins that other methods missed. It's like having a super efficient inventory system for our cells.
So quantum seems to really excel at analyzing like existing data.
It does.
But the paper also kind of hinted at its potential for tackling these different types of experiments.
Yeah.
There was something about label-free versus TMT-labeled proteomics.
Oh, right.
Can you shed some light on that for us?
Absolutely.
Okay.
So imagine you're organizing your closet.
Oh, okay.
Label-free proteomics is like identifying your clothes by sight.
Okay.
Their color, fabric, style,
Let's see.
TMT or tandem mass tags
Okay.
is like adding those little colored tags
Right.
to help you sort and categorize everything more efficiently.
So instead of just looking at the clothes themselves, we're kind of like using these tags to glean more information.
Precisely. These tags allow us to analyze multiple samples at once
Right.
and compare protein levels with greater accuracy.
Interesting. So quantums can handle both of these approaches,
Yes, it can.
making it even more versatile.
Exactly.
What are some of the advantages of using these TMT tags?
Well, one key advantage is the ability to achieve what we call absolute protein quantification.
Absolute.
Yeah, it's like getting an exact count of each item in your closet
Okay.
instead of just noting that you have, say, more shirts than pants.
Right. That makes a lot of sense.
Yeah.
And the paper mentioned that quantums actually performs like really well for this absolute quantification,
Yeah.
even compared to like other tools out there.
It does. They compared it to established tools.
Oh,
The
really?
Proteome Discover, and they found that quantums delivered exceptional results.
That's amazing.
And what's really exciting is that quantms is open source,
Oh, cool.
freely available for researchers worldwide.
So it's like sharing a secret recipe that everyone can benefit from.
Exactly. Democratizing scientific tools like this is crucial for progress.
Right. It takes a village.
It does. And it's not just about the software itself.
Yeah.
The quantms team is also advocating for standardized file formats.
Okay, so what does that mean, like, practically speaking?
So this ensures that data can be shared and reanalyzed seamlessly, right?
Okay.
So we avoid confusion and we foster collaboration.
It's like making sure that everyone is using the same measuring system.
Exactly.
Essential for building a solid foundation for
Exactly.
all this future research.
This emphasis on open access and standardization really underscores a crucial point. Quantums is not just a tool.
Yeah.
It represents a broader movement towards more transparent and reproducible research.
I like that.
And with its ability to analyze data from, various experiments, including those using TMT tags for absolute quantification,
Yeah.
quantums offers immense potential for unlocking even more insights from proteomics data.
It's amazing. So far we've covered, like, faster analysis. We've talked about new protein discoveries.
Right.
And even ways to potentially improve disease diagnosis and treatment, down the line.
Yeah, absolutely.
But it feels like there's still more to this story.
Yeah.
The paper also mentioned something about creating a baseline of protein expression. What exactly does that mean?
Ah, yes. That's where it gets even more intriguing.
Okay.
They used quantums to create what you might call a protein census.
At a protein census.
Yeah, like a reference map of normal protein levels across different tissues in the human body.
So it's like we're establishing a benchmark,
Exactly.
a standard to compare against when things go wrong.
Precisely. Having this baseline is crucial for understanding what changes when disease sets in.
Like having a before picture
Exactly.
to compare with an after picture, right?
Exactly.
It's a great way to visualize it.
Yeah.
Did they make any, like, surprising discoveries while they were establishing this baseline?
They did. For example, they found that some proteins that were previously thought to be specific to certain tissues
Okay.
are actually present in many others, just at lower levels.
Oh, wow. So it's like realizing that rare ingredient you thought you could only get at a specialty store
Right.
is actually available at your local supermarket.
Yeah.
Just, maybe in smaller quantities.
Yeah, it's an apt analogy. It challenges our assumptions about protein expression
Right.
and highlights the power of this kind of large-scale analysis.
Yeah.
This protein census offers a valuable reference point for future research,
Okay.
allowing scientists to investigate how protein levels fluctuate in various conditions and diseases.
It's like having this detailed map of a new territory
Yes.
that opens up, like, endless possibilities for exploration.
Indeed, this baseline data provides a foundation for understanding the complexities of the proteome and its role in health and disease.
Right.
And that brings us to a crucial question.
So we've explored this powerful new tool, quantums,
Right.
this
Yeah.
groundbreaking protein census.
Yeah.
But what does this all actually mean for our listener, sitting at home right now?
Right.
Why should they care about all of this?
That's the most crucial question. Ultimately, proteomics research like this has the potential to revolutionize healthcare.
Okay.
The deeper our understanding of proteins and their functions,
Yeah.
the better equipped we'll be to develop new therapies and personalized medicine approaches.
So we're not just talking about, like, abstract scientific concepts here.
No, not at all.
This research could have a direct impact on people's lives.
Absolutely. Imagine a future where we can diagnose diseases earlier,
Oh, wow.
develop more targeted treatments, and even predict individual responses to medications, all thanks to a deeper understanding of proteins.
So it's like tailoring healthcare to each person's, like, unique protein profile.
Precisely.
That's amazing.
And tools like quantums, along with, resources like this new baseline protein expression data, they're paving the way for that future.
It's mind-blowing to think that every cell in our bodies contains this complex world of proteins all working together to keep us going.
It really is.
And now we're getting even closer to understanding, like, their language.
And who knows what incredible breakthroughs await us as we continue to delve deeper into the proteome.
I'm excited to find out.
Me too.
Well, that's all the time we have for today's deep dive into the world of proteomics.
Fascinating discussion.
We hope this has given you a new appreciation for this incredible field and its potential to really change lives. Until next time, keep those questions coming.